Hi everyone, today we learn about RDNA technology. Today we learn about second year botany. It's a very important question. In every paper we can see this question. It's a very important question. Every student they have to be learn this question first. After that you have to go for any other question. This question there is no question paper, no doubt at all. These are tools of RDNA technology. In RDNA technology, process of RDNA technology, and tools of RDNA technology. From these two, one question is definite question. So we have to concentrate here. Then here simply 7 steps are there. If you learn these 7 steps, if you learn these 7 steps and how you learn, I will give some tips and some techniques also. How do you remember? I will give some mnemonics also. But you have to pay attention here. Let us enter into our class, that is RDNA technology. What is DNA technology? What is RDNA? Here he may ask like this. The question will be like this. Process of RDNA technology, process of biotechnology, process of recombination DNA technology. What are the steps involved in recombination DNA technology? He may ask like this. In that, you have to write these all the steps. Let us see one by one. Number one, first we have to learn where the DNA will be present. Everyone know the DNA is present in chromosomes. The chromosomes are present inside of the nucleus. The nucleus is present inside of the cell. If construct the structural and functional unit of the living things are cells, in the cell the key software, the key information may present on DNA. May present on DNA. Already we know DNA, the structure of DNA, that is double helix present in the DNA may present in chromosome. The chromosomes are present in nucleus. The nucleus may present in cell. My dear friends, deoxyribonucleic acid, the DNA may have information. That information we have to take that information and we have to insert into the another DNA. That is recombination DNA technology. Re means again. Combination means to combine two DNAs. That we call it as a recombinant DNA technology. Sir, what is necessity of recombination? My dear friends, for example, two desired characters are present in two different organisms are two different individuals. Look here, very simple. We have a desired character. That desired character, the desired character may present in the two different individuals. The two desired characters we have to see in one. That is one case. The second case, a gene is present. The gene product. Sir, what is gene product? Gene product in the sense the gene it consists a information in the form of codons. That codons it have a information. It produces some protein. It produces some protein depending upon the information may present on the DNA. Now information according to that The products, that is proteins, are produced here. The proteins are produced according to the information may present on the DNA. May present on the DNA. That DNA, it have information. So we require that information because of, for example, like that, which products we require. Which products we require, we produce by in vitro conditions, by in artificially. For that, we require this technology. That technology we call it as a recombinant DNA technology. Let us see how the recombinant, how the DNA we may cut and we may, which gene is present a DNA, that gene we require, that gene we have to cut it and we insert into the another gene. This is a DNA. On DNA we have a desired segment, desired fragment, desired piece. In that we have this information. We don't require all the information. At that time, how can we cut it and how can we paste into the another DNA? That technology we call it as a recombination technology. DNAs we have to insert, we have to merge, we have to cut and paste into the another DNA. That process we call it as a recombination DNA technology. My dear friends, we cannot see the DNA with our naked eye. It is a microscopic structure. We cannot see even cell. How can we see that DNA? So, in that case, we have to follow some procedure. What is that procedure? Let us see. My dear friends, what we are in this, we have to cut the DNA. First of all, we have to cut DNA. We want to cut the DNA. We require DNA, but the DNA is present. Where it present? Inside of the chromosome it is present. The chromosome is present inside of the nucleus. The nucleus is present inside of the cell. But how can we touch that gene? How can we touch that DNA? Here, very simple technique is there. We have to break down this cell and we have to break down this nuclear membrane. Then we have to break down the DNA. What is the process? Let us see. Okay. Okay. Then only we can touch the DNA. Let us see. Very simple steps are there in process. 7 steps are there in recombination DNA technology. The first step is isolation of DNA, isolation of genetic material, cutting of DNA segment at specific location, and isolation of desired DNA fragment, and insertion of isolated DNA into the suitable vector, and fifth one, introduction of recombinant DNA into the host cell, and sixth one, selection of transformed host cells. Next one, seventh one, obtaining the foreign gene product. What product we expected from that particular gene, that is the last step. First step, what is that? Isolation of genetic material. Genetic material is nothing but DNA. Here, mechanical method is there, by enzymatic method are there. Why? We have to isolate. Isolate means we have to take DNA from the cell. We have to obtain, we have to take it out DNA from the cell. For that, we have to follow a simple procedure. Very simple. The material is present inside of the cell, inside of the nucleus. For that, we have to break down all this cell wall or cell membrane, and nuclear or nuclear membrane, and chromosomal or which is the present unnecessary material may present in the nucleus, that also we have to remove. Then collect the DNA which we require. For that we follow these steps, very simple. For example, we have to break down the cell wall. If it is the plant cell, we are using some enzymes to break down the cell membrane. We want to break down the cell membrane. We cannot see with our naked eye that cell membrane, but we are using some enzymes. We are using some enzymes and we have to break down that. For example, it is plant cell, we are using cellulase. We are using cellulase. If it is the plant cell wall, the plant cell membrane is made up of cellulose. So, we are using, for break down the cellulose, we are using here cellulase is an enzyme. Like that, the bacterial cell, we have to break down the bacterial cell, then we have to use the lysozyme. Fungal cell, that fungal cell we break down by using the chitinase. You remember three things here. If it is the plant cell, cellulase. If it is the bacterial cell, lysozyme. If it is the fungal cell, it is chitinase. By different enzymes, depending upon the which source we are using. Okay. Like also we are using for break down the cell membrane and cell wall. Okay. Most of the times we are using enzymes. Then very fine. Now cell wall or cell membrane are breakdown. Then the nuclear membrane is there. Okay. The nuclear membrane is there. For breakdown of this nuclear membrane, we are using some nucleases. Then the nuclear membrane also break down. Then in this nucleus, we need only DNA. So here some RNA materials are also present in this nucleus. That is ribosomal RNAs are there. But we no need that RNA material. For that, we have to remove that RNA material also by using proteases. Then when we are using proteases, that RNA material also break down. My dear friends, now the chromosomes are present. In the chromosome, DNA is present. Okay. When we are using RNAs and protease, and by the protease, this which is affected, the DNA is surrounded by some proteins. That proteins also destroyed when we are using protease. Okay. Then the DNA is now present. The DNA may isolate. When we add cool ethanol or chilled ethanol, it will be the DNA precipitation will be comes out. Or DNA, we can be seeing that DNA in white precipitation or the thread-like structure we can find out when we add cool ethanol to that particular DNA material. Okay, my dear friends, in this, first step is isolation of genetic material or isolation of DNA. In that, the first step we have to break down cell wall or cell membrane. Depending upon which type of material we are using, that by that we can use enzyme like lysozyme, cellulase, chitinase. When we are using this, the cell wall will be or cell membrane will maybe break down. Then we are remove the RNA by using protease. RNA also remove. Now we have to isolate the DNA. Further, we add cool ethanol. Then it will be the precipitation. The DNA will be comes out. We can see it with our naked eye. We can spool it out. We can take it out by inoculation loop, or by we can use some material, by we can take it out by using the inoculation loop. This is the first step. I think you are understand very easily. Okay, let us enter into the second step, that is cutting of the DNA at a specific location. Now we have DNA. Look here, this is the DNA. But we don't need this total DNA. We required only a particular segment. For that, we have to cut the DNA. That is the second step. We have to cut the DNA. How can we cut the DNA? By scissor? No, we cannot cut. But we are using some enzymes. That enzymes we call it as a restriction endonucleases. Restriction endonucleases. The enzyme it cut nucleic material inside. So that enzymes we call it as a restriction endonucleases. It's a very important for the 2 marks also. Okay. That restriction endonucleases we also call it as molecular scissors. It cuts the molecules. It cuts the DNA molecule. So that we call it as molecular scissors. We call it as molecular scissors. Okay. Depending upon the where we have to cut, where we want to cut, depending upon that, different types of the restriction endonucleases are there. For example, EcoRI. This EcoRI is an enzyme. It obtained from the so that we give name EcoRI by using different restriction endonucleases. We cut the DNA into the small fragments. By it have, my dear friends, here a simple thing is there. Every restriction enzyme, it may cut at a specific site. It do not cut anyway. It cut a specific site. Let us learn third step. That is isolated DNA or desired DNA fragments. We have to cut a DNA, then we have to isolate that particular DNA segment by using the methods. That methods are gel cascade method or gel electrophoresis, and then we have to collect that southern blotting technique. Let us see what is gel electrophoresis. Look here, my dear friends, we have a cutted sample. Look here, in this test tube, we have all the DNA segments are cut in here. Already it is in the fragment stage. Then these fragments, we need only selected fragment. We need only desired segment. How can we select that desired segment? By using the gel electrophoresis. Gel electrophoresis means by using gel, we cascade the wanted DNA. Look here, we insert this sample into the gel it consists wells. It consists a well, and we have to treat this gel electrophoresis under the electric field. when that DNA segments or DNA fragments are moved according to their molecular gradient. Very simple, look here. Depending upon their size, the DNA molecules are moved according to their molecular size. It settled in different areas. Okay. Once again, these DNA segments are separated according to their molecular gradient. In electric field, we are supplied this gel electrophoresis means we supply positive charge and negative charge to this gel electrophoresis. Then according to their molecular gradient, the genes are moved. Then we take which gene we need by molecular weight or by selection of markers we have. Okay, we know known marks or known DNA segments by molecular weight. We can obtain that molecular gradient or molecular gradient separated DNAs by Southern blotting technique. Gel electrophoresis means very simple. We prepare the gel like a sheet. In that we keep wells on top. Then we apply current to this gel electrophoresis. We take a liquid EDTA. In this, due to that EDTA, it will be the molecules of DNA molecules or DNA segments are moved according to their molecular gradient. Then we isolate, then we obtain which segment we require. My dear friends, first step we isolate DNA from the cell. Second step, isolated DNA we have to cut into the small fragment by using restriction nucleases. Then we have to select which gene segment we require. That is the third step. And the fourth step, isolated DNA we insert into the suitable vector. This segment already we isolate. Look here. This is the DNA segment. Okay. We require this segment insert into the another DNA suitable vector. We have DNA. That with that DNA segment we cannot do anything. But when it express, it want to be express their information. When it express, when it is in the present in another host cell. For that, we have to insert this DNA segment into the another DNA vector. What is vector? Let us see the fourth step. Insertion of isolated DNA into the fourth step. Insert isolated DNA into the suitable vector. Vector means which gene segment, which material it can be transferred into the another living organism, that we call it as a vector. Here example for vectors, cosmids, plasmids and plasmids and bacteriophages we are using as a vectors. But we are using here vector is a plasmid. Look here. Very simple. Remember it. Plasmid. Different types of the plasmids are there. What is that? We will learn later. Plasmid means extra chromosomal DNA. It is a lower molecular weight it can be accept any DNA segment. Right, here DNA plasmid is there. Now, when we want to insert the DNA, before that we have to cut this plasmid. Am I right? Yes, we have to cut this DNA. Again we are using restriction enzyme. Which enzyme we are using at the time of the restriction endonuclease? What we are using for fragmentation, or what we are using the DNA cut into the fragments. Restriction endonuclease we are using here. Okay, then this plasmid will be cut. The plasmid will be cut. Then we have to insert our desired DNA into this plasmid. Into the plasmid. Then here plasmid is there. That wanted segment is there. Our desired DNA segment is there. Then we have to insert this DNA into the vector. That's the that is the fourth step. Now the vector will be there, vector and DNA. Now plasmid plus DNA, that our rDNA will be formed. Recombinant DNA will be formed here. That we call it as a chimeric DNA or rDNA. Why we are called it as rDNA? The plasmid is also a DNA. And DNA, our wanted segment is there. These two are combined to each other. That we call it as a desired DNA. That we call it as a recombinant DNA. We also call it as chimeric DNA. Now we have rDNA or chimeric DNA. Now we have to insert this rDNA into the another suitable host, because without host, with this rDNA we cannot do not be used. My dear friends, here one more technique also there. We have only single segment, am I right? Single desired DNA, but we have multiplied this DNA into the number of the DNAs, or we have rDNA. We have to manipulate, we have to duplicate, we have to Xerox this rDNA into the PCR. Okay, my dear friends, look here once. We have rDNA. With the single DNA we can't do anything. So we require number of the DNAs. For that we using PCR. That is polymer chain reaction. That machine we also call it as a thermocyclone. Okay, thermocyclone. Look here, that DNA, that recombinant DNA, we have to multiply number of the copies we can prepare by using PCR. Then that rDNA we have to insert into the another host cell, suitable host cell. That is the fifth step. We require suitable host cell, because when it is present in the suitable host, then only it express. Then only it will be expressed. For that we are using different techniques. That is the micro injection method, and second one is the gene gun method, third one is the heat shock. Look here, this is the host cell. Look here, this is the rDNA. This rDNA we have to insert into the another cell. For that we follow different techniques. We have rDNA, we insert into the another suitable host cell. Look here, this is rDNA, we insert into the another host cell. At that time we are following micro injection. We are in micro injection means, look here, this is the micro injection. By this injection we insert our RDNA into a cell that we call it as a suitable host cell. My dear friends, we are doing in this test tube, but we don't know which cells are transformed. Okay, what is transformed cell? Transformed cell means which host cell it consists plasmid, which host cell it may uptake RDNA, which host cell it accept RDNA, means which cell it consists RDNA inside of the host cell. That cells we call it as a transformed cells. It transformed with our RDNA. We are doing this experiment in test tube. In that we cannot see which cells are transformed, which cells are not transformed. For that we are using a technique, that is the sixth one, selection of transformed host cells. The transformed means which cells it consists our RDNA. That cells we call it as a transformed cells. That's transformed cells. We select transformed cells. For that we follow a method, that is the selection markers or colony hybridization. Sir, what is colony hybridization? Very simple. For example, look here. This is the already what we call it as, this is plasmid. This plasmid it consists ampicillin resistant sites and tetracycline resistant sites. Ampicillin resistant site and tetracycline resistant site. If it is the transformed, the cell is transformed when we culture the transformed cell on this cells on transfer. When we transfer this culture on media which consists antibiotics like ampicillin and tetracycline. The media it consists ampicillin and tetracycline resistant. The media it consists ampicillin and tetracycline antibiotics. When we transformed that culture on that media, the cells which are not transformed, the cells are died, it may not be grown. Because of they do not have resistant site, they do not have plasmid. They do not have plasmid means they do not have ampicillin resistant site, they do not have tetracycline resistant site. Which cells are transformed, that cells only can grow in that selection media. Okay, now the colonies we have to be cultivate, and we culture that colonies and we grow that microbes. Then we get product from that particular cells. That we call it as the sixth step, selection of transformed cells. Now we selected the transformed cells also. Then we use the transformed cells for production of what we required. Okay. Further, we are used, that is the seventh step, obtaining of foreign gene product. Which product we are expected? Product we are get by bioreactors. We call it as a obtaining of gene product, or we can also call it as a downstream process. Okay. These are the 7 steps. Very, very easy. Number 1. Isolation of DNA by using the different enzymes like lysosome, cellulose, chitinase. Then we are using protease. Then we are using cool ethanol. Then at the time we spool it out DNA. That's the first step. The second step, we have to cut the wanted DNA into the segments, into the fragments by using the restriction endonucleases. That is the second step. We have cut the DNA, we have to select the DNA. we have to select the DNA. That is the third step. Fragment wanted DNA we selected. That is the third step by using the gel electrophoresis. Then wanted DNA segment we have to insert into the suitable vector like plasmid. Okay. Now that we call it as a rDNA. That R will be multiplied by using the PCR. Then DNA we have to insert into the suitable host cell. Then we have to cultivate we have to select the transformed cells by using the selection media. Then we have in the bioreactor we can get byproducts what product we have. This is the rDNA technology. Okay, I think you are understand. We will learn in next class tools of rDNA technology. I hope you are understand. Subscribe our channel, like our channel. Thank you, one and all.